interrupt. Command.
Imagine this.
Request navigation.
Your aircraft has state-of-the-art avionics,
This is command. Stand by.
but your map is from the 15th century.
Command, please advise. Please advise.
Neuroscientists are stuck in this harrowing predicament as they try to make their way around the human brain.
The first classical map made about a century ago is still widely used today.
Published in 1909, it defined regions on the pinkish-gray organ that control our actions and functions.
The fundamental unit of brain organization for the cerebral cortex is what we call a cortical area.
So on the map, you could say there's a country called speech, another called short-term memory, another hand movement. Identifying every one of these cortical areas became a major objective of the Human Connectome Project, the HCP. Leading the effort, a master mapmaker.
I consider myself a cortical cartographer.
By July of last year, the HCP completed its first phase, a 21st century world map of the brain.
We reported the presence of 180 distinct cortical areas.
97 of them were new to brain science.
The Connectome gives us this opportunity, a really great tool to be able to navigate the human brain.
Navigating the brain is more than just naming cities and states. You need to understand the connections between them.
The word connectome implies that the fundamentally important thing about brains is connections.
This evening, we'll be talking and learning about two different kinds of connections.
Structural connections in the brain versus functional connections in the brain. By structural connections, we're really talking about the physical connections.
The wires that connect nerve cells.
So structural connectivity looks at the regions and the pathways connecting them.
The functional connectivity, though, is more about how different parts of the brain work together on an ongoing basis.
Brain function arises from conversations that different brain regions are having with each other.
A functional map must track living data, conversations flowing through the pathways between brain regions.
We collected data from 1,200 individuals. We have them do different tasks in the scanner, things like a memory task, emotions on faces. Those same regions of the brain seem to be forming networks.
Brain structure is wired by its experience, but we have no idea in what form that wiring diagram has that information.
And that's the big puzzle. To see how that translates into individual nerve connections, you need to look much closer than David or Deanna's technology can go.
Down to the level of individual neurons.
2 quadrillion times smaller.
What exactly is 2 quadrillion times smaller? Well, if the scale of David's brain map is the big picture,
Equivalent to a map of the whole Earth.
then the area that Jeff's covering would be a city about the size of, say, Sheboygan, Wisconsin.
That little blur, that little blob there might be Sheboygan, or we're not sure.
That little blob is a 1 millimeter cube of brain tissue, and it contains a staggering number of neurons.
50,000 nerve cells and about 1 billion synapses.
So right now, there's no way to model an entire human brain by diagramming its micro-wiring. Scientifically, for now, we're lost in the foggy limits of brain science.
You would need more digital information than is the actual digital content of the world.
Hello, everyone. How would you like to do a program after Tracy Day's daughter said happy birthday to you? I mean, I'm just going to weep here for 90 minutes. That was so sweet. And thank you so much, Tracy and Brian and their whole family that's created this wonderful festival. And wow, I just couldn't be more grateful. And, you know, she comes out here every time and thanks the sponsors. And, you know, I just want to reiterate, I mean, for 10 years, these sponsors, I mean, I don't get paid for this festival. We wouldn't think of it. I mean, there is so little money that goes to the what you see that's not absolute, just like right on, hardcore, unfiltered, this is the real deal science. There's nothing like this in the world. So I guess that's why they called it the World Science Festival. That was really smart of them. But in terms of just the payoff versus the pay-in, this is something really unusual. Secondly, I would like to say that unlike a lot of the programs that I've done on Sunday, we are actually saving the best for last today. This is a riot of a show. I mean, you're just not going to believe this show. First of all, how many of you heard that last line of the film? It was kind of, it's easy to lose stuff like that, but did you hear what he was saying? Yeah, okay, so let me repeat it for you because it is absolutely staggering. The millimeter, the cubic millimeter of brain tissue, if we were to just model that, okay, including all the synapses and their connections and their capabilities of having conversations and communications with each other, it would require, listen carefully, more digital content than exists at any given moment on Earth. That's everything. That's like all the Googles every day, constantly, everything. That's just insane. That is just an insane task. And these scientists are like, we're ready to go. Let's take it on. I mean, that is more complicated than anything I can even imagine. But wow. How many of you saw the movie Fantastic Voyage when you were, like this big? And a really cheesy science fiction movie where they, get into a submarine and go into human beings. And I think it's been referenced on this stage before, but the denouement of the movie is they go into the human brain and they cut out this clot that's in the human brain, and they're sailing around the density that was in that cubic millimeter. I mean, these guys were just sailing through the brain. Oh, there's a neuron over there. Oh, there's one over there. Oh, there's the clot. And then they zap it and save the day. But this idea of going into the brain and knowing where you are and having a map of the brain was so fascinating to me as a little kid. Wow, because the brain, is not just the thing that you see, with the wrinkles and stuff. It's so much more than that. And then, we're talking about static maps. We have to remember that the brain is place plus function times individuals, okay? So a map of the brain would have to account for your propensity to eat Butterfingers candy bars, and your propensity to watch Robot. It's not Robot. No, it's I Robot. Or your propensity to watch old episodes of How I Met Your Mother. I mean it would just be so unbelievably complicated because it's dynamic, it's moving, it's not static. There's nothing about the brain that is static. The rest of the body is mechanical, maybe electromechanical, maybe electrochemical mechanical, and there'll be a bunch of scientists that come out here and just beat me up and say, we are very complex too, okay, buddy? But I mean, neuroscience, oof. There's another idea here that I have in terms of mapping, and I'm just wondering if is there anyone in the audience who knows what this says? Call it out. Put the slide up. Anybody? What's the word? What's the translation? You don't know the translation? In English? Truth? Think. It's the word that IBM used. All IBM employees had on their desks, okay, a little thing like this that said "think" on it in the language of the employees. Where are you from? Pakistan. Okay. So put the map up. So this is IBM in Urdu, and from the 1960s. It was a cooperative effort to think, to try to understand digitally what thinking means, what processing means, what data processing means. And the map part of this that I love is the word think is trying to describe what the brain does, right? And IBM is this company that's trying to figure out how to make machines that think. And it's got people all over the world. And so then there's like the word for think all over the world, so you can map the word onto the idea, onto the company, onto the machine, onto the brain. It's maps everywhere you go when you think about the brain. All it is maps. And that's so fantastic to think about. And for the rest of this presentation, this segment, we are going to have the most wonderful scientists in the world talk to us about their experience with mapping, with map making, with map conceptualizing, with brain understanding. We got the rock stars here, folks. Our first guest is a professor of neurobiology at Washington University in St. Louis, principal investigator for the Human Connectome Project, a fellow of the American Association for the Advancement of Science. He's internationally known for his research on the structure, function, connectivity, evolution, and development of cerebral cortex in humans and non-human primates. Please welcome David Van Essen. Our next participant is a professor of psychological and brain sciences at Washington University, St. Louis, and an investigator with the Human Connectome Project. She's on the scientific board of the Brain and Behavior Research Foundation and the Stanley Foundation, a fellow of the Association for Psychological Science, and a member of the American College of Neuropsychopharmacology. Please welcome Deanna Barch. Now, Deanna, it is the case that the word neuropsychopharmacology is an example of how the sciences are kind of coming together, right? Mm-hmm. It didn't exist, probably 20 years ago, right?
Well, 50 years ago.
50 years ago, okay, all right, date me, thank you, that's good, excellent. All right, joining us is a professor of molecular and cellular biology and the Ramona Cajal Professor of the Faculty of Arts and Sciences at Harvard University, developmental neurobiologist, member of the National Academy of Sciences, Jeff Lichtman. Our final guest is an associate professor of psychology at Columbia University and director of the Developmental Affective Neuroscience Laboratory. She's a recipient of the National Institute of Mental Health Biobehavioral Research Awards for Innovative New Scientists, the American Psychological Association's Distinguished Scientific Award for Early Career Contributions to Psychology, and the Developmental Science Early Career Researcher Prize, NIM Tottenham. So, we had that little film there. I came out here and, was kind of a smartass. Anything you want to take issue with in terms of any of the science, any of the comparisons, any of the metaphors that have already been bounced around here at all? You're cool. You're comfortable so far.
You hit the nail on the head.
What?
You hit it on the head.
Hit it on the head. Hit the nail on the head, which of you hits nails on the head?
Every day.
Every day, that's right. Great. I would love to know what the Connectome is. It's a federal project. It's one of these, global, everyone's cooperating project. Jeff, what's the Connectome?
Well, on this stage you have 4 people who would probably have slightly different ideas about what the Connectome is.
That's what we like.
Yeah. So from my perspective, the Connectome is a project to map the connections between nerve cells at synapses, at the level of synapses, which is doable, but the price we pay is this extraordinary amount of data we need, so that doing a cubic millimeter is for us a triumph, and we're not done with a cubic millimeter, I should say. That's 2,000 terabytes of raw data.
That's not a zip drive. That's,
right.
Yeah.
But in terms of the other realms, and maybe you guys can talk about this, a cubic millimeter is your voxel size. That's the smallest thing you ever see.
Nim, what do you how would you describe the Connectome Project to someone who asks you, so what do you do at a party?
Well, I think building on that answer, there's so many levels to address when you're thinking about the connections in the brain. So there's the very, very small levels that have tremendous meaning and contain an incredible amount of information. But then you can also look at more macro levels that we can use with fMRI that tell us about a different level of representation of information within the brain. So it's going to take a lot of data to be able to map out all of those various levels.
And Deanna, well, how would you describe the Connectome?
Well, so I think I would agree with Nim in the sense that there's really connectomes at many levels of analysis, at the level that Jeff is working on, and then at the level that says, okay, all those different cubic millimeters in the brain, how are they wired up and how do they work together, and how do those conversations, to use a term that Nim used, kind of support all the different behaviors and thinking and emotions that humans are able to do. So I think we really need all of those across all of those scales eventually to really understand the full human connectome.
And David, would it be fair to say, and I deliberately left you last here, that you're the most macro here?
I think it's fair to say that Deanna and Nim and I all work at the very macroscopic level of the luxury of working on the entire brain of living individuals. And the other part of what drives us is the need for not only as much information as we can, but as rich across different types of imaging. Magnetic resonance imaging is the dominant one that we'll talk about here, but there are also other what we call modalities, magnetoencephalography, electroencephalography, give richer information in the rapid time course of the signals they follow, but not as good in the terms of the resolution in space. So all of these types of information can work hand in glove to give us deeper insights into an incredibly challenging and exciting set of problems.
I think we could say the turnout here suggests that there is a broad consensus that we want to know a lot more about the brain, the Human Connectome Project. If that is the pathway to get there, then we really, really support it. But just for the hell of it, can you convey, without betraying too much proprietary information or making enemies at all, where would the disagreement be of the four of you here? Who would be inclined to most disagree with the others? All right, let me help. David, raise your hand. Who would disagree with David scientifically? That's a nobody. Okay. Deanna, raise your hand. Who would have a responsible scientific argument with Deanna from time to time? Okay. And Jeff, raise your hand. Who would have a disagreement scientifically with Jeff?
Well, I don't know. I would say, I don't know.
Okay.
I don't know that we would disagree per se, but we may put a different emphasis on what might be critical levels of analysis.
Right. I was hoping we weren't just dealing with chicken brains here on the stage. Nim, where would you say, in terms of the two parts that we're going to be discussing, functional and structural sort of aspects of the macro brain, where are the scientific disagreements within that? What are the arguments? What are the big questions that would be analogous to cosmologists saying string theory, other cosmologists say, oh, that's a bunch of hogwash? It's not as intense as that, I know, but there are disagreements, right?
Yeah, I mean, I think when just looking at the people that we have on the panel here, one question that we want to know is, are the data that we're getting at the level that Jeff's seeing comparable to the types of level that we're getting with fMRI? And what is fMRI measuring?
What does that stand for, fMRI?
Functional magnetic resonance imaging. So
and what's that taking a picture of?
It's the data that gives you the activation patterns on the brain. So what we commonly think of as the blobs on the brain.
And is that the pictures of the neuron electricity?
So it's not electricity. It is an area of active inquiry to try and understand what is the bold signal. So when you're seeing those colored pictures, what you're getting is a statistical map of a signal called BOLD, which is giving us an index of where oxygen is being distributed differentially across the brain as the brain is engaging in a particular task.
So it's data that presumes that a mapping of blood activity and oxygenated blood activity in the brain is comparable to neuronal and cognitive, no, maybe not cognitive, that's too much to say, but electrochemical activity in the brain. Is that fair to say?
Right, it's an inference. We can
Right.
only make an inference with those data.
How good is that inference, Jeff?
The challenge is in one little blob of the fMRI image, you have 50,000 nerve cells and 1 billion synapses from hundreds of different cell types over many different layers of cerebral cortex. Each part of the cerebral cortex is layered, and there are different kinds of things going on in each layer, and all that is compacted into a single dot. And
On an fMRI image?
yes,
Yeah.
so it is a global average. I would say if you tried to do the equivalent of an fMRI on the United States, you would be looking at energy use in cities, perhaps, and you would
Sheboygan?
find, yeah,
Since
Sheboygan,
we're
maybe
trashing
that's right.
Sheboygan
You
for
might
some
be able to
reason.
get a little bit of Sheboygan data. You'd get more data from bigger cities, and these cities have a lot of energy use in the daytime. They fall off at night, but it's not like nothing is going on. But you would not be looking at each individual person or each individual activity. You would be looking at, essentially all the lights on in a particular part of the brain at a particular time. And so you can infer something. I'm not saying you can't infer something, but there is still a chasm, I think, between the machine that's doing the work, which is very, very intricate, and these rather global macroscopic techniques that can work in a living human brain. If you want me to do this to your brain, I have to kill you. So
Yeah, I'm scared of you most of all, Jeff, that's for sure. Deanna, fMRI, useful tool, but there's a huge horizon beyond it. That's fair to say?
Yes, absolutely.
And David, you would go along with that? We don't need to belabor this point.
I also agree strongly, and I would point out that there's another type of information that is very important for the Human Connectome Project and other endeavors, which is we call diffusion imaging and looking at structural connectivity. And that is also a very informative method. It relies on the preferred diffusion of water along the length of axons within the white matter is a little bit faster than trying to get across the membranes of each axon. That indirectly informs us about local orientations of fiber bundles that can then be stitched together to estimate long-distance connections from one region of gray matter to another. That is also fraught with its own challenges because it's looking at bundles that are crisscrossing one another to reflect the actual reality of brain circuitry, and we can make estimates, but they're not perfect. And so another of the hot debates in the field is what can we infer that we can put confidence in, and what do we have to be very careful of and not overinterpret?
But I have to say, David, those are some damn pretty pictures. I have to say, I love those. Those are great. I want some of those. All right. Let's ask one other question. The Connectome. It's a project that's partially government funded or wholly government funded?
The Human Connectome Project was funded by the National Institutes of Health, NIH, over the period of 2010 to 2016. So it's actually wrapped up and achieved its major aims, and it was publicly funded through the National Institutes of Health. There's a follow-up set of projects that are looking at across the human lifespan, again in healthy individuals, looking at babies and children and older adults, again publicly, federally funded to make it happen.
Any sense of the vulnerability of those projects going forward, Jeff?
Yeah, I wanted to add something kind of interesting, that the high-level maps are being funded through a part of the government called IARPA, which is a relative of DARPA, but instead of the D for defense, the I is for intelligence. So
they're not exactly opposites, but...
yeah. So the government is interested, I think, from health reasons, the National Institutes of Health, but also for many other reasons into getting a better sense of this map at many levels of resolution. I think thinking that there may be some deep insights into how brains do things so well that computers at the moment do less well, or at least less efficiently.
This is a bipartisan project, and it would appear to be a consensus initiative, a scientific initiative in Washington. Is there any obvious reason to think that it's going to go away in the next 12 months?
I mean, I don't think there's necessarily an obvious reason. The current administration has proposed a significant cut to the funding for the National Institutes of Health, but we know that there's strong bipartisan support for the kind of research funded by the National Institute of Health because it has such dramatic implications for understanding human health and biology and defense systems and other things. So I think the hope is that that strong bipartisan support will continue.
And
so
will be a pushback.
And will be a
Yeah.
pushback.
How many of you wanted to know the answer to that question out here? Oh, okay, all right. Now, for those of you who are watching us in the stream, the hashtag to send tweets and questions and to further this discussion, which has a little bit of disagreement to it, if you'd like, is hashtag WSF17. I've got an iPad right here, right there, actually, and I can ask your question as it happens. So feel free, start right now. We won't wait till the end on this one. All right, so let's take a look at some of the structural issues here, which are maybe some of the more familiar kinds of things. David, who inspired you to do this work, and what was your original conception of a brain map?
I've been fascinated by the cerebral cortex for 40 years, and when I first started working on the problem, I was looking at the visual cortex of an animal model, the macaque monkey. It has a sheet of tissue that is convoluted to get it to fit inside the skull of a monkey. And one of the challenges I faced was that in order to get information about the brain and the connections, it was necessary to take slices of the brain and look one slice at a time. I was compelled by the analogy with maps of the Earth that two-dimensional maps of a convoluted sheet are very informative. So at that time, I actually used pencil and tracing paper to make crude but useful maps of the visual cortex and then the entire cerebral cortex of the macaque monkey. I knew this was a job better suited for a computer, and when I moved to Caltech in 1976, we started working on that problem. But it literally took 2 decades to get the computer horsepower, but more importantly, the algorithms into operational shapes for others to lead the way. A number of groups have worked on that effort. So now we can make computerized maps of the monkey cerebral cortex, but more excitingly, of the human cerebral cortex, which is even more complicated and convoluted.
When you were kicking around back as a advanced science student in high school and maybe college, what were the visuals that you could see that would show you what the map of the brain or the functionality of the brain supposedly was all about?
They were pictures in books, and it's nice that you brought forward Exhibit A, which is the classic, century-old map of the human cerebral cortex by the great German anatomist Corbinian Brodmann.
This is the 1909 map you were talking about in the film.
That's exactly right. I learned about it in college, and it's still taught today as one representation, but it's analogous to maps of the Earth's surface from the mid-16th century, for instance, which were quaint and colorful. Brodmann, there's a colored version of his, but this
probably maybe get it. Go keep talking.
This is the map that he used, and it was actually accurate to this day for a few of the 50 regions that he identified, but there was reason to suspect that this was not the whole story, and it's been a century-long endeavor to make better maps of the human cerebral cortex.
Die Rindenfelderung der lateralen und medialen Hemisphärenfläche des Menschen.
Your German is impeccable.
Mittellang 1907. So getting from these maps you had to, and you alluded to it a moment ago, really begin to describe the meaning of the folds in the brain, right?
That's correct. I think of the folds of the human cerebral cortex as analogous to the crumpling of the Earth's surface into mountains and valleys and other geographic features. And that's, of course, very important. It's part of the physical substrate for understanding the lay of the land, quite literally. But we also care intensely about the functional organization of what's happening on the Earth's surface, and those we call political subdivisions, countries and states. We know that there are about 200 countries of political subdivisions, and many of those respect the boundaries imposed by rivers and mountains, so there is some correlation between political subdivisions and the geographic features on the Earth.
Oh, and some of them believe in climate change, and others don't.
Right.
Right. Yeah.
And these social and cultural inclinations, dispositions reflect the interactions and communications amongst the billions of people that make up the population of the Earth. In like manner, the cerebral cortex contains about 16 billion neurons in each individual, and it's the interactions and communications, the local and long-distance connections and functions of those neurons that determine what we call the parcels or the cortical areas in the human cortical sheet.
So is it the case that in the beginning, the brain is smooth in an embryo?
In
and
an
then
embryo,
development,
and even into the first 2 trimesters of human embryonic fetal development, it's very smooth, just gets slightly curved in its shape.
All right, so this is first trimester, just
Right.
like this. So no real crinkling
So
there at all.
by the beginning of the second trimester, things have gotten into somewhat curved shape. But in the explosion of cortical growth in the third trimester of human gestation, things get really wild and it crumples up like that. Yours is good for one individual, and mine is even more crumpled.
So it's
pretty severe, yes.
So like,
and so by
and the
birth,
more of this, the more of this.
Well, the more that can fit inside the skull that has to get through the birth canal. That's one of the reasons why we have to have a highly convoluted cortex to get a pizza-like sheet of tissue for each hemisphere, crumpled up to fit inside a compact skull that can get through the birth canal. But by birth, the cortical convolutions are nearly as complex as they are in an adult, even though the brain is only 1/3 the size of an adult brain.
Okay, so baby up to about age of 6, and then from 6, it
6 months to 9 months.
no, but from 6 years.
Oh, then
it begins to
it gets bigger.
like age 60.
Well, then things start going south, yes,
Yeah.
not north. we can sympathize.
Happy birthday, John. Yeah. Great. All right, terrific. That was my best present of the day, I have to say. All right, but it only goes to show how important the brain is in terms of creating more complexity in a confined space, right? You're limited in the space. You're unlimited in the level of complexity that you can do. There are other limitations, but this is how the brain solves its own sort of problem of dynamics. You discovered a cortex, right? Isn't that true? That was missing from one of the old maps?
Well, we in the process of using data from the Human Connectome Project to get more accurate maps, Matt Glasser, my very talented student, and I and other colleagues in our consortium used multiple types of information, what we call multimodal mapping, to make a more accurate map than Brodmann had provided us that identified 180 areas in the left hemisphere and a matching set, remarkably symmetrical actually, in the right hemisphere. And one of those particular areas that we were fascinated by was our poster child area called Area 55B. And it's a little tiny area that's related to language processing, and it's one part of an entire language network. But it had been overlooked in most studies. We called it Area 55B because it was first identified in
New Mexico, right? With aliens.
Pretty close. Pretty close.
yeah.
It was discovered in Germany by another great German anatomist. And it wasn't a highway.
You're the best. Go ahead, and keep going.
And so Hopf had described this area as a region that had a little less myelin than its neighbors, and that's all he had to go by, but he was right on target, we believe, for that area. But now we have a more accurate mapping and a much better signature of at least some aspects of what this particular little area, 55B, is doing in conjunction with a network of language-related areas.
Let's get a couple of questions here from the people who are watching our stream. And this is one that came right at the beginning. Why spend so much time and money mapping every single connection if we might never be able to finish it? Jeff? Well, it is for you.
You know, I think everyone probably would agree that the Human Genome Project was worthwhile. This was a descriptive attempt to get the nucleotide sequence down to every single nucleotide in the DNA of a human being. And when that project was first proposed, there were a lot of people who said, why should we spend all this money to get all this DNA? We know a lot of it is junk DNA. There's a lot of things in there we don't care about. Why do this? This is just going to be impossible to understand. But no one in the scientific community, at least, doubts the extraordinary value of actually having a data set that describes the entire human genome. And not only that, now that we have the way to do it, we can get your genome and my genome, and they're different. And the genome of people who have diseases are a little different. A profound amount of insight has come from generating that data set. And I see connectomics just like I see genomics. It is a way to describe the brain at enough resolution that we can begin to make reasonable hypotheses about how brains of people who have disease are different from brains of people who don't have diseases of the brain, for example. How baby brains are wired differently than adult brains. Everyone knows a newborn baby is not speaking, not walking. We're just blobs of protoplasm, can't even turn over.
Except for the ones on the Upper West Side.
Except on the Upper West Side, right. Yeah. But, you know, a lot must be going on. And old people, even old people who don't have dementia, and my children would count me in this category, are a little different from young people. We're a little more set in our ways. Why is that? Where does that change take place? Where are all our memories stored? There are millions and millions of questions, I think, where this kind of data would be justifiable.
And again, one could conclude, well, it's cultural because the older people do more authoritative stuff, so they have to be set in their ways. But in fact, it could be purely biological, and then the biology informs the culture, and then the culture, and again, the map goes on, and the sort of fascinating interplay is exactly what we're discussing here. I'm gonna dream about that tonight now. Nim, would you say that the maps that we have now, the visual sort of tools that we have now, are helping us to pose new questions or answer more questions than they pose?
Both, for sure. I mean, I think that as a developmental psychologist, the reason why I came into this is because I was very interested in behavior and where behavior comes from. And being able to get some tool that gives us structure and function to attach these behaviors to changes everything. It starts to answer the question that you raised, which is, how does the environment get under the skin, so to speak, right? So to use an analogy from the heart, Our cardiac tissue is the tissue, and our heartbeat is the behavior that's associated with that tissue. So likewise, our brain is the tissue, and behavior is the output of that tissue. So if you want to understand and be able to ask deeper questions about behaviors and where they come from, and why am I different from you, and why are we similar in other ways, then being able to have this tool changes the game.
And because we were describing, and you did that very eloquent defense of genetic information, but again, there's this huge threshold of disconnect between genetic data and neuronal data and behavioral data and what the brain functionally does. Deanna, can you explain for us the great pictures that we have of the identical twin brains? Vin, go ahead and put the first brain up here. We've got brains of twins, I believe here. Pictures of brains of twins. Okay, these are identical twins.
Well, I think that the twin data actually does a wonderful job of illustrating what Nim talked about in terms of both how the environment gets into the brain and how genetics shape the brain. So when you look at the brains of twins, there are things, especially identical twins who share the vast majority, 99% of their genetic makeup, there are clear similarities, things that are more similar about those twins than other ones. But there are also fascinating differences.
They don't look identical. Identical is not a word I would even begin to use.
Because there are also environmental factors that get under the skin and into the brain that are going to shape how the brain wires up, how those convolutions are created over time, and the twins' behavior will probably follow and be similarly different or the same depending on how much they've experienced the environment differently.
And even if you take identical twins, I don't would actually do this, and put them in identical green rooms and fed them identical green food and gave them identical glasses of water at the exact same time every day for 8 years, you would still get differences.
You would get some differences for sure, yeah.
But it would be less.
It might be. That's actually a great question. We don't know.
It's not a good experiment, though, right?
We're not going to do that experiment.
Thank you. Yeah.
But I think, obviously to the degree that we believe environment also shapes brain development, the more similar the environment, the more likely it is that, those are going to be shaped in a similar way. But again, we can't really do that experiment to deliberately manipulate how similar or not similar the environments are.
Go, David.
But since most of the cortical folding, the development of the convolutions occurs before birth, that's actually occurring in not quite identical, but in the same womb and very similar environmental influences. So the fact that the outcome at birth of newborn identical twins, their brains are very different, that tells us that a lot of this is driven by subtle, perhaps epigenetic influences, but and perhaps small differences in environment lead to perturbations that lead to different folding outcomes. But the other part of what's important, I think, here is that even though the newborn twins or any individual has a highly convoluted and relatively mature-looking cortex, during the expansion that occurs by a factor of 3, especially in early after-birth postnatal development, the regions that change the most are the regions that we associated with higher cognitive function. So that's where our plasticity, our ability to learn and develop and emerge with sophisticated concepts like drive this conversation, that's occurring in specialized regions of cortex that are more plastic. They are also more vulnerable to abnormal experience and perturbed development. So this relationship to disorders and disease comes into play in that regard as well.
So during this rapid period, things such as the autism spectrum might emerge because it is such a complicated and rapid developmental period that is not well understood physiologically.
That's correct, although we also know that there is a genetic component, a heritability to autism in terms of the likelihood of having an individual, a child, somewhere on that autism spectrum disorder. but it's called a spectrum for good reasons, because there's tremendous diversity in many
Right.
ways, and that's where the experience, particularly in early childhood development, is likely to shape the trajectory of individuals, and where we desperately want to understand how can we decipher what's going on and perhaps better treat and modify the outcome.
There are four, I guarantee you, burning questions here, probably for each of you. But before we get to those, Nim, can you describe, let's extend this developmental discussion, because a clock begins, as David was sort of saying, at birth, and there are various kinds of developmental pathways that are emerging at different times, and we've started to separate them a little bit and really understand what race takes place at what time.
Yeah, so human brain development is one of the most fascinating things, right? Because we start off, all of us, as a single-celled organism, and then we all go through this miracle of development where we create the most complex 3 pounds in the universe. And we all do it more or less the same, and yet we all have beautiful individual differences. So where do those come from? So what people have discovered, which I think is just a beautiful design of the human brain, is that when we're young, our brains overshoot. They overproduce cells, they overproduce synapses. So once upon a time, when we were young children, we had many more cells in our brain with many more connections, and it was a much denser forest than it is now. So why would Mother Nature go through that whole process? Because that's really inefficient. Metabolically, that's incredibly, arguably inefficient. And the reason is because that gives us what David referred to earlier, of this incredible plasticity during development. So it's like throwing a big fishing net out into the ocean and then pulling back what you need. Because when we're new on this planet, when we just land on this planet, we don't know what household we're going to land in. We don't know what the climate's going to be. We don't know what the language is that we're going to have to learn. And yet we all do it. And so in many ways, in order to understand the mature human brain, you want to understand brain during infancy and childhood, because that's what gives us that end product where we're all exquisitely adapted to our environments in which our brains grew up.
But the profile of the adaptation process, the becoming an adult, the maturation, the development of personality, et cetera, is actually a subtractive process.
It's a series of progressive and subtractive or regressive processes. So if we look in the back of the brain where the visual cortex is, you see this overshoot that I described, and then this pruning away, like you prune your rose bushes, pruning away of the synapses that your brain learns it doesn't need. It's
So
inefficient to keep those.
What are the two functions here that are being looked at?
So in the pinkish line, we're looking in the back of the brain in the
Okay.
visual cortex, and you see that initial increase in synaptic density, of the density of the connections, and then you get this pruning back as we get older. And then if you look at that blue line, that's the frontal cortex in the front of the brain, and it does the same thing, but you can see that the whole curve is shifted over. So that's reflecting that we have these processes of increase in synapses and regression that's happening in our brain, but in a hierarchical fashion where we go from the back of the brain to the front of the brain, which really reflects the way that these behaviors emerge over development, right?
Professor
so
Nim?
we can, yeah.
Does this mean that evolutionarily, cognitively, we require visual information to inform our cognitive selves, and so that's why cognitive development might lag behind visual?
Right, so brain development makes sense evolutionarily, but also within one individual's lifetime. So first you have your sensory systems coming online because you need to be able to take in information from the environment. Then you've got motor systems to allow you to interact with the environment. And those are necessary prerequisites in order to do all of the cool higher level stuff that we can do, like plan for the future or fall in love or regulate our emotions.
Or even be blind. I mean, blind people are going through an adaptive process.
Right, so we have multiple sensory systems, right? So we have this plasticity early in life where we can shift resources where it suits us best.
Gosh, that's great stuff. All right, from, this is, again, related to the initiatives that have gone on really since, long before 1909 to try to figure out what's going on in the brain. And in our lifetime, I think it's fair to say we've taken, at various periods of time, a sort of moonshot approach to either early childhood education or, let's put the Mozart on the navel of the pregnant ladies and let's make sure we can possibly intervene in these great neuroplasticity moments and create great humans, better humans. Here is a question from one of those moonshot kinds of projects. Sally James, Twitter user, how much has the Allen Institute brain map helped you all, if at all?
So I just returned from a trip to Seattle to be a participant on the advisory board for the Allen Institute, and I've gotten a window into their bold, privately funded efforts to map.
Paul Allen.
Paul Allen, co-founder of Microsoft, has been incredibly generous over approximately 15 years in investing in privately funded neuroscience research, aiming to do at his Allen Brain Institute things that are not readily done in a university laboratory context. So that started with a gene expression atlas in mice. The field has benefited greatly from. It's migrated to the Human Gene Expression Atlas that again is a unique resource that I've gained from by comparing gene expression patterns to brain networks, functional connectivity, and lo and behold, they are correlated in interesting ways. The Allen Brain Institute is now moving on to look at structure and function and connectivity and wiring in mice and in humans, and is continuing to carve out a niche that I think serves the scientific community very well, again, through this find an opportunity that fits their model and their agenda.
All on the same team, we would agree.
Yes.
Yeah. All right. So here's a question from, and this relates to what you said earlier, Jeff. And Jack Hederi is the user here. By analogy to the genome, since we were talking about that just a moment ago, how much individual variability do you expect in the connectome? Does that question make sense?
Yes, it's
Okay.
a great question.
All right.
Obviously, there are 5 of us sitting here, and we all have an entirely different set of experiences that have molded us into the adults we are. People like me believe that information is embedded in our wiring diagrams, so it would be shocking if our wiring diagrams are very similar. Now, it's true also that genomes are not identical, otherwise we would all look the same, which would be scary, I think.
But there's no way in which the connectome variability would be on the order of chimps have 99% of the same genetic information that it appears that humans do.
I think this is a good question. It may be depending on what level of resolution you look at. If you look at the major pathways that connect parts of the brain, things that come out of the bold technique, and perhaps out of diffusion tensor imaging, you might find that brains are 95% similar. I actually don't know, but probably they're quite similar. If you get down to the wiring of a little piece of cerebral cortex, my guess is that you will find substantial differences between the brains of different people.
Deanna.
Well, I was going to say, it's kind of the glass half empty, glass half full, and I think this is one of the really interesting things that's coming out of the data produced by the Human Connectome Project, because one of our explicit goals was to look at individual differences in brains and relate them to behavior. And I think we're seeing exactly that there is remarkable similarity in the sort of basic network structure at the level of Sheboygan and, in Chicago and New York. But there are also really interesting individual differences that seem to relate to behavior, including a lot of those sort of high-level cognitive and emotional and, motivational behaviors. So I think we're going to see it's both, that there's real commonality in key structures, but really important individual differences that link to behavior.
I think a lot of people would have trouble, or would not have any trouble distinguishing Sheboygan from Chicago or New York. Just even though I understand the similarities that you're talking about there, but again, it's a resolution question.
I think many people use this as a strong argument against doing this. Well, if everybody's brain is different, what are you going to learn by doing one brain? And it would be like saying, what could you learn about if every city is different?
Or why teach all the kids,
Yeah.
Really?
Right.
Yeah.
There's another example. But, every city is different. But if you studied in detail Paris, I suspect you would get insights that would translate to London and to New York.
And to Pittsburgh.
And to Pittsburgh.
Good one.
And to Oakland, and to Cleveland. Game 2, 90 minutes. On a somber note, at the resolution that you expect someday to be able to observe, would the kinds of macro social impacts of the tragedies in London over the last few days be reflected in the brain activity of virtually everyone in London who probably feels targeted at this moment? Mm-hmm. Would you expect that there would be evidence of sort of fear and that their brains would be changed after the event?
Of course.
Yeah.
What else? I mean, if they remember these events, and almost certainly they will never forget these events, where else would it be except in the brain?
I wasn't saying that it was going to be in the foot, but all I'm saying is that everyone in London would have some impact. We like to think in terms of proximity. Obviously, people who lost individuals and who are grieving are in a special category, but the entire, to have something happen on a scale of a city is go ahead, David. Rescue
but it
me,
also
please.
speaks to the profound diversity in how and whether memories are stored in vivid ways. So people will remember those events in London if they intensely experience them. I remember as an undergraduate student learning in a chemistry class in Caltech of the shooting of John F. Kennedy. That's seared into my memory. There's a ton of other things that I simply don't recall about my early years. So the way in which memories are encoded and retracted or regained is a fascinating question for neuroscience and why there's so much tremendous interest in the nature of memory encoding, because it's not just remembering 1% of what happened yesterday and 0.1% the day before. It's very specialized in ways that are important for our understanding human behavior in society.
No, and some of the best sessions here at the World Science Festival have been about memory and the Candel work that's been done and all that great. So could I ask the four of you to stand up, because it seems to me that you demonstrate something that is very rare, I think, in science. You, Deanna, and Nim are structural. You study structural or functional?
We're more functional.
Functional. Okay, so why don't you go stand next to Nim? Yeah, yep. Go ahead and stand next to Nim. Yeah, that's right. And Jeff, you and David are more. You seem to be much more functional. Or are you structural? No. Structural. Okay. All right. So stand next to David. All right. So these are two very different disciplines with a very similar mission, and as you saw at the beginning of the film, they're hugely far apart in terms of the tools that are necessary to make progress in these various disciplines. I've done so many sessions here at the World Science Festival where the differences and the different groups and the different people, I mean, we could put the chairs like 3 times the distance between them. That's how far removed they are. But you're actually working very much together. I mean, these disciplines have to, so even this distance is incorrect. I mean, you would have to, I mean, you're hugging, right? Isn't that true? What does that look like? What does that actually look like? That's like, see.
We got it.
So that, you may sit down. That is the level of cross-disciplinary cooperation, and you're seeing it tangibly there. You have no idea how hard it was for me to get them to do that. You have no idea. Actually, it wasn't that hard at all, but this idea of combining brains and brain science and missions of what it is we should be studying and trying to figure out, and what is complex, what's a hard problem, what's an easy problem. It's so great to see people working together like this, and this is what science is all about. All right, let's have a couple of stream questions here. Oh, we got tons of time. You'll love this. There's always this one, right? From Facebook, James Cunico. Is free will an illusion? In other words, be nice. Be nice. He's just sitting at home in his shorts on the computer. There's so many other things he could be doing. Is free will an illusion? In other words, is our brain the be-all and end? Now this is sophisticated. The be-all and end-all to our decision-making. When you get to the connectome, will you be looking at a deterministic image? Anybody want to take that on?
I would like to, because one of the great surprises for me when we began actually reconstructing brains at the level of resolution of the wiring diagram is the insane density of wires and synapses in the brain. It might as well be called free will because it is so darn complicated. And so it's not that you have to find that one place where there's a switch that you're going to throw dice and you're going to go left or right. The whole brain is made up of, billions of elements, trillions of synapses actually, that each have a certain amount of stochastic behavior. So you might as well call this free will. It is free will for us. We believe we have free will. If you look down from an airplane at New York City at night, you'll see most people on the highways are leaving the city. You look early in the morning, most people are coming in. That doesn't seem like free will to somebody up there. It seems very much like we're doing exactly what we're supposed to do to make the city work. And I think maybe brains are sort of like that.
And if you do that in North Korea, I mean, it's really, not looking like free will. But I've never heard that. I've never heard that expression before. The idea that the very complexity itself departs from, it becomes autonomy. That's really amazing. I can't tell you how many stoned arguments I've had. It was just a complete waste of time in my life. That's brilliant. Thank you for that. That's another birthday present. I wasn't even expecting. Anybody else have to say anything on free will?
I agree with Jeff. It is just, it's in our control. What does control mean? That's part of what we want to decipher. Will we ever understand it? Perhaps not. But fundamentally, the richness of brain circuitry gives us, in practical terms, the free will that is our handle on that concept.
All right, this is another one that we always get in sessions like this, and it's a good one, and it's very well stated. Alan Vincenzi, who's a YouTube user, do you believe brain data, including but not limited to the connectome, can be faithfully recreated in a von Neumann machine? Now, a von Neumann machine is something like a Turing machine, a theoretical idea of a computer that would reach a level of complexity that would begin to behave comparably to what we think of as autonomous units. That's a very primitive way of explaining what the von Neumann machine is, and I'm sure we'll be getting some emails from Alan Vincenzi later on today. But anyway, just for you so you know what that is all about, anybody want to comment?
Again, I get this question a lot, that if you get the wiring diagram, aren't you 90% of the way there to having a thinking machine? The wiring diagram is a static entity at the moment you take it, and it doesn't tell you the way information is flowing through those wires. This was one of the great discoveries of neurobiology, this idea that information flows in a directional way through individual nerve cells. That information is often stimulated by things from the outside that are activating your retina or the cells in your ear or your skin. And none of that is built into these wiring diagrams. That's coming from the outside. And so when we look at the wiring diagram, we don't see what is actually going on. At the level of fMRI, at least you see areas of the brain lighting up. But at this fine level of the way the machine works, we're far from having a way of putting information into those wires in a way that simulates the way actual experience goes into the brain.
Deanna, let me see if I can get you to comment on something that Nim said earlier. She talked about the wide net, that developmentally we produce a lot more synapses than we apparently need, and then there's some sort of essentially subtractive process that gets us down to what we can actually use or, will work for us or something like that. That would suggest that the level of complexity, if I can call it the Lichtman threshold, that turns a wiring diagram inspired machine into an autonomous machine is actually much lower than what you would need if you were trying to specify exactly what a human being does. I mean, crickets are autonomous in a certain sense. I mean, you don't. So this idea of where the threshold of autonomy might lie on the von Neumann threshold or the Lichtman threshold. I mean, is this nonsense, or would there actually be a threshold of complexity that at some point we may discover?
I mean, you raise a good question. I do not think Westworld is going to evolve in the next month or so. It may be that there are some organisms where the complexity is such that we will actually be able to sort of recreate their functions in a machine. If you look at an amoeba or something where we may be able to map it out and make it work. But, with humans, the degree of complexity that's present even from birth and even for relatively simple functions is such that I think we may be able to do approximations that may engage in behaviors that look on the surface similar to what humans do, but may not be arising through the same mechanisms. And what I think I have a hard time, and AI has struggled with this too, is humans' complete ability to encounter new things and be flexible and to, and one of the great things about, babies as they develop, they start to be able to take a face and recognize that same face even when they've never ever seen that particular view of it, right? Or that coloring or with a hat on and the hair, and can recognize it. That has been incredibly hard for machines to be able to recreate. So I guess I'm kind of with Jeff that I think where it's hard for me to imagine that we will have a machine that can recreate the full plethora of complexity that we have in humans.
But we're failing at one-celled creatures at this point too.
Yeah.
So, Nim, you have a book there, and Jeff, I'll get to you in a minute. I think it was there.
I don't have the book.
Somebody strike it.
I had a book.
You had a book. You had a book from the 1930s that had some fantastic pictures of neurons.
Yeah.
Which, you're kind of in love with the neuron itself as a functional device that accomplishes so much in our own brains. Can you describe how you were inspired by some of the original visual
Yeah, I would say
pictures?
I'm in love with the developing neuron. So that book was published in 1939 and contains some of the images that we showed earlier of the changing density in synapses across age. And so, the work that we're doing in development is sitting on a long history of people being fascinated with how there's my book. Yeah, thank you.
I mean, what's so great about this is that we knew there was an image that you've used in your work that came from a book that nobody could find. Our producer, Vin, found the book.
Yeah.
And I mean, like, for producers, ladies and gentlemen, number one, I mean, consider how many terrorized synapses it took to find this book.
Yeah.
Anyway, go ahead.
He's a really good producer. So,
no, hold it open. I think wherever you want. Just to
So this brings me back to our first discussion about the chasm in between, the type of data that Jeff's lab can get versus those from an MRI lab. But so that might leave you feeling a little sad about what we can do with imaging. But I want to point out that between this type of book being published in 1939, there wasn't a whole lot more that we could do in terms of understanding brain function in a developing human who was not ill until the 90s. So the first paper that was published using functional MRI in healthy, awake children was in 1995 by B.J. Casey, which is really not that long ago. And so it's just a testament to how young this science is, but how much has happened. So it's a very exciting time in developmental psychology or developmental neuroscience with regard to humans.
And here's a question directly to that. Brendan O'Hara says, how does learning multiple languages early on affect brain development? Any knowledge about that?
Yeah, there's a lot of excitement in this area. So one just speaks to the behavior of language, right? So your brain will undergo neural Darwinism during development. It learns based on who's being activated, which synapses are being activated or not. If they're being activated, then I'm going to keep them, and if they're not, then I'm going to prune them away. So if I'm being exposed to both English and Japanese as a 6-month-old, then my brain is going to hang on to both of those.
Is that your experience? Is that your experience?
That was not, no, I don't speak Japanese. I wish. That in and of itself is amazing, that we're that plastic, right? So I can't speak Japanese. There are many sounds, phonemes within Japanese that I will never be able to process. But when I was 5 months old, I could. But my brain decided it didn't need to hang on to those anymore. So that in and of itself is amazing. And then the second thing is there are all these cascading functions, all these cascading benefits that you get. So children who can speak two languages fluently also have this amazing benefit in their prefrontal cortex. So they're able to switch very easily in terms of cognitive functions because they've had a lifetime of switching what languages they're speaking and understanding what the other person is expecting of them in terms of language.
I've also heard, and this is somebody from South Asia who spoke, I think, 7 languages. Her experience was that once you get past 2, 3, 4, and 5 are easy. Mm-hmm. Does the brain, is that what the brain tells you?
Right, so the brain starts picking up on what are the common principles that I need to extract from a language. And so as you gather, I guess 3, 4, if that's the magic number, then you get a broader sense of what are the generalized principles here that you can apply to new languages. So yeah, I would say probably the second language is the hardest to learn, and after that, it's like, it's nothing to brag about.
Your brain's just feasting on all this.
It's nothing to brag about after that, right?
Oh.
John, one thing that's important about that too is those effects don't just last through childhood. Mm-hmm. There's evidence that they last your entire life and actually may be protective later in life against things like cognitive decline and even dementia. More work is needed there, but there's a lot of evidence that they have very beneficial, long-lasting effects.
David, you were going to say something, and I interrupted you, and then we started onto this neuronal romp here. Do you remember what it is you were...
It was a follow-up to the question about autonomous agents and artificial intelligence. And one thing I've learned from over my scientific career is, never say never too earnestly, because some of the things that we're talking about as everyday scientific experiences were out of the realm of what I could even imagine, let alone say that would be possible someday. So technology advances are driven by the laws of physics, but they can make profound advances.
So some of what we were talking about up here right now were impossible to imagine when you were an undergraduate.
That is correct. Even magnetic resonance imaging. I remember thinking about, would there be some way to measure brain activity, and talking to experts, and it didn't seem like it was going to go anywhere in the early 1980s. That was lack of vision and insight about how the clever ways into the new technologies could be achieved by physicists and engineers. But I was also going to come back to the point about artificial intelligence, and many people know about entities like Watson and other extraordinarily sophisticated computers, which in some contexts can do impressively well in communicating with humans and giving the appearance of intelligent thought. And arguably there is some degree of intelligence there. I don't think Watson now would be able to sit at this with a fifth chair or sixth chair and carry his or her digital weight in this kind of free-flowing conversation.
But I heard Watson described one time as a combination of the Library of Congress and sprinter Usain Bolt. Running around getting source material.
Working in a profoundly different way. So will there be versions of artificial intelligence in the future that are dramatically better than Watson? Unquestionably, in my mind. Will that ever approach the cleverness and insight and perspective of real human beings having a conversation like this? I personally am on the skeptical side. I wouldn't say impossible, but I'm doubtful. And I think if they are doing it, one of the fascinating challenges is to what degree will computers emulate human-like thought strategies? To what extent will they use principles of neural circuitry like Jeff focuses on to process information in ways that emulate the characteristics that Deanna and Nim and I look at in the living human brain? A huge, open, and exciting question, and it'll be up for the next generation of young scientists to carry that ball forward.
The remaining questions we have online also go to how I wanted to conclude this session, and that's with you, Jeff. I mean, in many ways, you're the astronaut in the suit, sort of untethered from the mothership, way out there. What inspired you to do the work that you do?
You know, the history of neurobiology began with this guy, Cajal, who made this rather remarkable leap of faith from looking at structure that has more or less stood the test of time, this idea of a directional network. And I think just as going from Gregor Mendel and his peas to the atomic structure of DNA to the human genome, going down and then back up again, I think neurobiology has to do the same thing. We have these ideas that now nerve cells are connected in a wiring diagram. That's what Cajal said. And now it's up to our generation and the generations following to get those wiring diagrams, to really know what they are. And that is what has motivated me. It just seems like a very logical next step in neurobiology, to not just say, oh yes, there's a wiring diagram, but what is it? Just like, oh yes, there is a genome, but what is it? I think that's what I'm after.
I think we have Cajal.
Yeah, I think something.
In the old days, some nice black and white.
Many people would say the birth of neuroscience came with the absolutely groundbreaking work of Santiago Ramon y Cajal, a Spanish physician, pathologist, and ultimately neuroscientist, who used a technique called the Golgi stain that allowed Cajal to stain brains in ways that very small numbers of nerve cells would be rendered dark and all the other cells would be clear. Cajal was a gifted visual genius. He had ink, a paintbrush, and that's what he did. He just drew pictures on paper. They are absolutely beautiful. And not only beautiful, but he inferred this very dynamic view of the way information flows. There's a network of cells, and the cells are of various types, and they talk to each other. And he was right, almost always. And it was such a radical idea that Golgi, who invented the stain, absolutely did not believe that that was true. Even at the Nobel Prize, Golgi kept saying that Cajal was an idiot, that he just screwed this up entirely. But time has allowed him to win out here. Yeah.
This is emotional for you. I mean, his commitment, the tools that he used, he understood they were primitive. He just went for it anyway. I mean, I just have to say that is such a beautiful story, and that you're carrying on his work. You are badass.
I mean, it's inevitable that this work would be carried on.
Well, let's see the tools before you downplay everything. Let's see the tools that you use to get beyond.
Okay. So the first thing we tried is if the Golgi technique is good and every small subset of cells are labeled black and you can learn something about the circuits, what if we could see every single cell? And so instead of black, we made every cell a different color. And we called this technique Brainbow, and it's been extraordinarily popular as screensavers for those. Yeah. So it's a very powerful technique of labeling every cell a different color, and in certain parts of the nervous system, it really does reveal circuits. But when you get to the cerebral cortex, guess what? There's just so many wires there that you can't actually tell the colors apart, because even at the finest level of resolution of the light microscope, there's a picture of those bigger blobs or nerve cells, and all that stuff in between, that felt work, are the gazillion wires that attach them, and you can't resolve them as individuals. So yeah, we have to go to a different technique. So that's plan A. So now plan B is what we're working on now, which is to do this not with light and color, but use electrons, which have a much shorter wavelength and give us the ability to resolve every one of these wires. And the way we do this is we take a block of brain and we fill it with a metal called osmium, and we take that block of brain filled with osmium, that little black spot in there.
This is why you'd have to kill me.
Yes, that's right. You have to take that out of your head. And then we put it in a plastic, very hard plastic, and we put it on a chuck, and we move it up and down against a diamond knife. That's what's going on in that second picture. And then from there, out comes one section after another. They're about 1,000th as thick as a human hair. So we get 1,000 per hair thickness. And then we pick them up on a tape, and then we take a picture of each and every one of them with a very high-resolution microscope. And the pictures are 250,000 pixels by 250,000 pixels, each image, and then we have to take 33,333 sections like that to do a cubic millimeter. So that's why
So you take that level of resolution in 2D and do 33,333 to make it 3D.
Right. So it's basically a film strip, and you take those pictures and you stack them up, and you get a stack of images that are now a three-dimensional version of the brain, and then you color in the objects using deep convolutional neural nets to generate the structures in the brain. Here is a dendrite of a nerve cell and an axon
Get
talking
out.
to that cell in green. And that's one little piece of an area that we did that is just so discouraging. It's so depressing. There's just so much stuff in these brains. There's 1,500 nerve cells. This is 3 billionths of a mouse brain. 3 billion. It took us 5 years to do 3 billionths of a mouse brain when we started. It's just extraordinary. And a synapse every cubic micron, so there are 1,500 synapses in there. Yeah.
I mean, I could say this to all four of you, but my response is just, I am not worthy. I am not worthy. I mean, do you ever think about how, 50 years from now, 100 years from now, they will look back on this work and...
As primitive and stupid.
It's
like the early days of genomics when people actually used their thumbs and pipette men to get the DNA sequence of a virus. And now we have these robots who can do a whole genome in a day or two for a few $1,000s. We are now back at the virus stage. We're just starting.
How old are you?
Unfortunately, too old.
But we can. How's the freezing thing going?
Well, some of the people who I've worked with, some of my colleagues, younger colleagues, are motivated by this idea that we have to get our wiring diagram into silicon so that we can store it and perhaps send it out into deep space at the speed of light, and maybe some advanced culture somewhere else will upload that or download it and then upload us somewhere else, that's the best hope for space travel. Yeah.
That's fabulous. I love that.
Yeah.
I mean, my head wants to go, but my body says no.
But I have to kill you to do this. I just...
No, right, of course. Why is it so short? Why is it so short? Final thoughts, Nim? On anything we've been talking about? Anything you'd want?
I'm still
our
stunned
audience?
from that video.
Yeah.
Hard to come back from that.
Well, I could throw one of these. All right. Here's one. Well, we all respond to this one question from our stream audience. How can we continue to develop new synaptic responses as an adult? Yeah. That's the kind of, Jack LaLanne question,
So we are. I mean, the fact that we can learn new things, the fact that we can form new memories depends on these microstructural changes happening. Otherwise, we would have no memory. So we have incredible plasticity early in life. It decreases, but it never goes away permanently. And some argue that in some areas of the prefrontal cortex, we actually never reach our full mature state because we're constantly exhibiting high levels of plasticity. So, and some of the exciting interventions are things like, how can exercise boost that even further for individuals who are experiencing cognitive decline? Or how can certain pharmacologic agents boost that plasticity further?
Career-changing millennials.
Career-changing millennials. Sleep. It's that all the things that we know are good for us are probably having an effect on increasing our plasticity.
Deanna?
Yeah, I was just going to comment on that, sleep and, there's fascinating theories about how sleep is a way to sort of clean up synapses so that you can do more the next day. There's fascinating work looking at brain injury and ways that rehabilitation can help with, rewiring the brain. So I think there's a lot of hope that it will continue on. It's less, but it still continues.
Jeff?
Well, I hate to say this because it's a downer, but being an old person, and I'd like to think that I'm infinitely flexible, but I remember when I was young being impressed at how narrow-minded my parents were when they were my age. And I have a feeling my children think a little bit about me this way. So although I feel plastic, I think the world is beginning to assume I just always talk about the same thing over and over again. I say it's because it's really interesting, but it's probably because all the other things my circuits could have done have been pruned away. So.
Rust. All we are is rust. But I mean, if you took up the violin or something, or you,
I wouldn't.
You wouldn't. I'm so glad I didn't end on Jeff. David.
Well, I see a lot of younger faces out in the audience, and I would reiterate the point that you guys are the future. Those of us who are above 50, to put it mildly, we will continue to work hard and look for opportunities to strengthen in plasticity, in healthy aging. We will continue to chip away at the profound impact of brain disorders and diseases, which is a scourge of society, and we desperately need better solutions. And we are excited by the interest in the brain and neuroscience that's manifested by the wonderful attendance at this session. And we look forward to future years and reading and learning about the accomplishment of the next generation. So thanks to all.
Well, I want to first of all thank all of you for participating in this 10th year of the Science Festival. I want to thank our great panel who participated. And when all else fails, think, okay? Stand up, get some more synapses. Woo!